Hi class, this is your blood analysis lab PowerPoint. This will be in PhysioEx exercise 11, and you'll need to complete the activities in that exercise 11 in PhysioEx on Hematocrit, which is activity 1, Hemoglobin activity 3, and blood typing activity 4. You can do those activities and upload them as files separately to the assignments or the submission tab under the modules. You'll also need to complete parts 1 to 3 of blood typing. There's 3 PDF files that I uploaded to the module, and you'll also need to do those. And this PowerPoint will take you through just an introduction to blood analysis. So the components of blood, if we take a sample of your blood right now, we call that whole blood. It can be centrifuged, and that means it can be spun down incredibly fast to reveal 3 components. When whole blood is spun very quickly, the 3 components separate into erythrocytes, which are red blood cells. Those make up about 44% of the blood sample. They'll be on the bottom of the sample. The buffy coat will be a middle layer containing all your leukocytes, which are white blood cells, which help in the immune response, and platelets, which help with blood clotting. And that forms about 1% of a blood sample. And then the plasma will be the straw-colored liquid that makes up the upper about 55% of blood. The erythrocytes, leukocytes, and platelets are what we call as the formed elements. So this is how we would take a blood sample, we would centrifuge it, and then the parts of the blood would be separated with the plasma on top. Plasma is mostly water, proteins, and other solutes. The buffy coat there you can see are your platelets, your leukocytes, and then the erythrocytes would kind of sludge down to the bottom, and those are your red blood cells. A little bit more about the functions of blood. It's the big transporter in your body, so it transports oxygen, carbon dioxide, hormones, nutrients, wastes. It helps to regulate things in your body like temperature, pH, and fluid levels. And it also works with protections. Your leukocytes, which are white blood cells, they guard against infection by triggering an immune response to antigens, which are substances perceived as foreign. Your blood also carries antibodies that will attack specific antigens. The formed elements consist of erythrocytes, leukocytes, and platelets, and these formed elements can be viewed with a blood smear. We won't be doing a blood smear in lab, but if you would withdraw a little piece of your blood after a finger prick, we could stain it and see the different types of blood. In the blood smear, we mostly see red blood cells. Anything darker colored, usually in purple, that would have granules or not have granules are leukocytes. And you might see a teeny tiny platelet in there as well. Out of the formed elements in blood, we have what we call is the hematocrit, which you will be learning more about in this lab. This is the percentage of the volume of all formed elements in the blood. Clinically, it is percentage volume of erythrocytes only, and the hematocrit will vary with age. Children can be a little more variable, and also sex. Males have a slightly higher, on average, hematocrit. And you'll be learning more about hematocrit in one of your activities in PhysioEx. This table shows you the diameter and size of each cell. So the erythrocytes, the red blood cells, have the largest diameter. It tells you their function, the lifespan, which is interesting to see. And then the density, the average number per millimeter cubed of blood between females and males. So erythrocytes are commonly called red blood cells. They're not true cells because they lack a nucleus and organelles. They're about 7.5 micrometers in diameter. They're what we call biconcave disc shaped, so they're indented on both sides. Their whole structure is designed so that they can carry as much hemoglobin as possible. That's why they don't have a nucleus or any other organelles. And hemoglobin is the protein that carries oxygen in the red blood cell. As they pass through teeny tiny small blood vessels, they line up in a single file termed a rouleau. And this rouleau can be seen here on the right, just a single file of red blood cells. You can see the biconcave shape of the red blood cell here, how it's indented on each side. Hemoglobin is what carries oxygen and carbon dioxide in the body. Every erythrocyte contains about 280 million molecules of hemoglobin, and each molecule of hemoglobin can contain 4 oxygen molecules. Hemoglobin transports oxygen and carbon dioxide. We call it oxygenated when hemoglobin is maximally loaded with oxygen, and deoxygenated when some oxygen is lost and carbon dioxide is gained. Each hemoglobin molecule consists of 4 globin polypeptide chains, 2 alpha and 2 beta chains. This is the structure of what hemoglobin looks like. There's a heme group containing an iron right in the middle, and it's just interesting to see these 4 chains, and they'll each carry an oxygen molecule. So each of the 4 globin chains possess the heme group, and thus each hemoglobin molecule can bind a total of 4 oxygen molecules. So again, each red blood cell has about 280 million hemoglobin. So if we multiply that by 4, you're talking about a trillion oxygen molecules per hemoglobin, which is an incredible amount. Now we get to the fun part, blood types. And this is probably one of the most confusing subjects that you'll probably talk about. So hopefully Dr. Tydell will come talk about this even more, and you'll learn more about this in lab, because I'm going to give you a quick rundown on blood types. How many of you know your blood type? I'm A positive. My doctor just told me that the other day when I was getting tested because I'm pregnant, so I just had my blood drawn. So how do we get these different blood types? Well, erythrocytes, those are the red blood cells, they contain surface antigens, and these are proteins or sugars that are attached directly to the red blood cell, and they are in the ABO blood group. The presence or absence of A and B surface antigens determine the ABO blood type. So for example, if you're type B blood, that means your red blood cells have antigen B only. If you're type O blood, your red blood cells don't have antigen A and they don't have antigen B attached to them. If you're type A red blood cells, or if you're type A blood, your red blood cells have type A antigen on them. So I am A positive, so I have type A surface antigens on my red blood cells. The specific surface antigens present are also accompanied in the plasma by antibodies. These are proteins in your immune system that will attack antigens that would be foreign to that individual. So if you're type B blood, you'll have anti-A antibodies in your plasma. If you're type O blood, you will have both anti-A and anti-B antibodies in your plasma. And if mismatched blood is transfused, the recipient's antibodies cause agglutination of the donated erythrocytes, and that can lead to hemolysis or splitting of your clumped erythrocytes. So this slide is explaining a little bit more about these ABO blood types. Let's just go through each one itself. So if you have type A blood, that means that your erythrocytes have the surface antigen A on them, directly attached to the surface. It also means that in your plasma, you're creating antibodies against B. So you have what we call anti-B antibodies. And you can see here how this picture is representing how these antibodies will fit or be able to touch or be the same shape as surface antigen Bs that are found in type B blood. That means that if type B blood mixes with type A blood, the antibodies in the type A blood will bind to those surface antigen B proteins and cause what we call clumping or agglutination of the blood. That is not good. That's a sign of a bad blood transfusion. If you're type B blood, you have surface antigen B on the surface, And in your plasma, you have antibodies against A. So you can see here that anti-A antibodies have a shape that will fit the surface antigen A's perfectly, so that if type A blood mixes with type B, your anti-A antibodies will clump with the surface antigen A's. That'll cause a clumping of your blood. It's never good when your blood clumps because you always want your blood to be flowing smoothly. There is a type AB blood type. Maybe some of you are type AB blood. That's where you have both surface antigens, A and B, on the surface of your red blood cells. Because you have both surface antigens, your plasma will not make any antibodies that will attack either one of the surface antigens that are on itself. So if you're type AB blood, you have both surface antigens on the blood cell surface, and you have neither antibodies in the plasma. It's good to be type AB because that means you can accept blood from anybody. We'll talk about the positive and negative in a second. If you're type O blood, that means you have neither surface antigen A or B, and because you have neither, you will create both types of antibodies against them in your blood. Type O blood is what we call the universal donor because it creates both antibodies in the plasma. It will be compatible. Then we get to what we call Rh blood types, and this is what gives you the positive or negative factor. So I told you I was A positive. Some of you might be B negative or O negative. What the positive means, that positive symbol just represents the presence of another surface antigen, and that's surface antigen D. So again, whether you're positive or negative, that just represents the presence of another surface antigen on your red blood cell. I'm A positive, so I have surface antigen A on my red blood cells, and I have surface antigen D. In your plasma, I do not create any anti-D antibodies because I'm not going to create antibodies against the antigen that I already have. However, if you are Rh negative, you don't have the surface antigen D, you will not create any anti-D antibodies. So unlike types A and B, which already have the antibodies in the plasma, if you're Rh negative, you won't create anti-D antibodies unless you're exposed to Rh positive blood. And this can be a problem. If you ever are exposed to Rh positive blood, you will then have those antibodies in your system, so that if you're ever exposed to Rh positive blood again, there could be a complication. This especially plays a role in pregnancy, which we'll talk about. So what happens when blood transfusions go wrong? We have what we call agglutination when there's a bad blood transfusion. Let's look at a good blood transfusion first. Donor blood type here is type A blood of donor, and type A blood of the recipient contains anti-B antibodies. So we're mixing A blood with A blood between a donor and a recipient. Because there are no antibodies against the type A antigens, we don't get any agglutination because these antibodies only will bind to anti-B antibodies. When there's no agglutination, no clumping between an antibody and the surface antigens on the red blood cell, you don't see any clumping in the sample. And this is what you guys will go through in lab. So this is what we call a successful blood fusion match. If we mix type A blood of a donor with type B blood of a recipient, remember that type B blood has anti-A antibodies in its plasma. And you can see here how the antibodies in the type B blood will fit really nicely and be able to grab a hold of those type A antigens. When it does that, it causes clumping, and the clumping can be seen here, literal clumping of red blood cells in that sample. So a little more about blood types. The Rh blood type is determined by the presence or absence of that Rh factor, which is surface antigen D, on the erythrocytes. When present, an individual is Rh positive. Like me, I'm A positive, so I have the A surface antigen and the Rh antigen. Anti-D antibodies only occur when an Rh negative individual is exposed to Rh positive blood. And an individual with all three surface red blood cell antigens will have A, will have B, and will be positive. So we call that person AB positive blood. So how is this a problem in pregnancy? And you'll answer several questions on pregnancy on your blood typing 3 activity, which is uploaded under this module. Let's say we have an Rh negative mother who wouldn't normally have any of those anti-Rh antibodies in her blood, but she has an Rh positive baby in her womb, which probably got from the father. When she has that Rh positive body in her womb, that will cause her plasma to create antibodies against the Rh factor in the baby's blood. This might not be a problem for her first pregnancy, although they still check that, but it can cause complications for future Rh positive pregnancies because now that mother has antibodies against the Rh factor in her blood, so she could be harming the fetus growing inside of her because now she has those anti-D antibodies in the blood that she was exposed to from her first pregnancy. I'll end with this slide because this shows you the blood type, who you can give it to, and who you can receive it from. And I will leave it at this. If you're blood type A positive, you can give blood to anyone with the same antigens or more. So let's say you can donate blood. This is a good way of remembering it, and Dr. Tydell will probably teach this to you too. But if you have a certain blood type, you can donate to anyone with the same antigens or more, and you can receive blood from anyone with the same antigens or fewer. If you look here, you can see that AB positive is what we call the universal recipient because it has all antigens on its surface, the A, the B, and the Rh factor. It won't create any antibodies against it, so because it has no antibodies in its plasma, it can receive blood from anyone. We call O negative the universal donor because it has no antigens on its surface. It can be given to anyone because anyone will accept blood with no antigens on the surface because the antibodies that they would have against it wouldn't be able to grab hold of anything. So I'll finish with that. Good luck with the activities. I hope you guys are all staying healthy and well.